Greetings to all. I am K. Thangaranjitham from Government College of Engineering, Tirunelveli. I am handling basics of biomedical instrumentation subject through this video class. Our subject code is OMD 551. Today we are going to see about the second unit, second topic, that is how EEG will be taken. So EEG is the electroencephalogram. Electroencephalography deals with the recording and study of electrical activity of the brain. By means of electrodes attached to the skull of the patient, the brain waves can be picked. The human brain measures about 1500 cm³ in volume and weighs 1.36 kilograms. It is placed inside the cranium, which gives its complete protection. The brain is covered by three membranes, namely pyometer, arachnoid, and durameter. The space between the pyometer and arachnoid is filled with the cereporous spinal fluid. The brain is divided into three parts, namely first one is forebrain, and then midbrain, and then hindbrain. So the figure shows the entire human brain arrangement. The different parts are cerebellum, medulla, lumbar, and these are all the topics we already studied in +2. So main parts of a brain are cerebellum, cerebrum, medulla, and sonal cord. Then how the brain activity will be measured, we are going to see about. So the brain activity is to be measured using EEG 1020 electrode system. This is the pictorial representation for 1020 electrode system. 1020 electrode system. Here the electrodes are placed in 10% and 20% distance as shown in figure. Here the first electrode is placed 10% and the next electrode will be placed apart from 10% and then 20, 20 and then 10. Depending upon our requirement, the electrodes are placed in 10 and 20% displacement. So now we are going to see about the working of 1020 electrode system. The commonly used electrode system for recording EEG signal is termed as 1020 electrode system or managed electrode system. The electrodes are placed at a distance of 10% and 20% appropriately from the total distance between the extreme endpoints of the skull, namely appropriately from the total distance between the extreme endpoints of the skull and the right and left ear lobes. This type of system is otherwise called as managed electrode system. In managed system, the electrodes are placed systematically on both sides of the skull. Both bipolar and unipolar system can be used. In unipolar, the differences in potential between two neighboring electrodes are considered. For unipolar, the reference electrodes are placed on the non-active part of the head like forehead or on the ear lobes. First, the distance between nasation and enions is determined and the distance between right and left ear lobe is also determined. The central electrode is placed at the intersection of the imaginary lines joining nasation and enion and the right ear and the left ear. This is the experimental procedure to measure the 1020 electrode system. And then this is the overall EEG electrode recording setup. Here the skull, here we are placing 1020 electrode system from which the channels will be selected, that is which electrode is going to transmit the signal is to be selected by channel selector, and then the signal will be amplified through this amplifier circuit, and then after filtering the harmonics present in the particular signal, and then it will be recorded using this recorder arrangement. The trigger signal gives the corresponding trigger to turn on the amplifier, that is once the triggering signal give the trigger and then the amplifier give the signal, pass the signal to filter. From filter we can able to record. So here the audio stimulus, visual stimulus is to trigger the signal processing triggering circuit. EEG recording setup includes the patient cable consists of 20 electrodes and is connected to the right channel selector. Every channel of the channel selector consists of an individual multi-stage AC coupled differential adjustable gain amplifier. These amplifiers must have right gain and low noise characteristics since the EEG potential are in microvolt range. The amplified EEG signal are passed through the filter bank. Typical cutoff frequencies for the low pass filters are 5.3, 1.6, 0.53 and 0.16 Hz. Band pass filters tuned for 60 Hz and used to eliminate mains frequency interference. The high pass filters have typical cutoff frequencies 15, 30, 70, 300 Hz. The output voltage from the amplifier may either be applied directly to the 8-channel display through the filter bank, or it may be stored as data on a tape recorder or computer memory for further processing. This is the entire analysis of ECG waveform. This EEG waveform. This EEG waveform is usually taken normally occipitally patient and the normally patient or frontally. This is the waveform for children and sleeping adults. This is the waveform for premature babies, infant and sleeping adults. This is the different condition we are going to measure EEG, that is sleeping condition, normal condition and abnormal condition. These waves are known as alpha waves, beta waves, theta waves and delta waves. And then similarly we are going to measure the EEG in awake condition, light sleep and depth sleep condition and cerebral death condition and epilepsy. So for different condition we are going to find out the EEG waveform. From this analyze with reference waveform, our brain also compared with reference waveform, and if there is any malfunction present in our brain, it will be calculated through this arrangement. This is known as EEG 1020 electrode setup system. Thank you dear students.